The discovery of King Richard III's remains has rewritten history in ways no one could have imagined. For more than 500 years, his final resting place was a mystery. Chronicles told us he was killed at the Battle of Bosworth in 1485, the last great fight of the Wars of the Roses. His body, according to reports, was taken to Leicester and buried without ceremony in the Grey Friars Priory. But time changed everything. The friary was dissolved, its buildings destroyed, and its exact location lost to memory. Some later rumors even suggested that his bones had been dug up and thrown into a river. For centuries, many believed the grave of England's last Plantagenet king was lost forever. That belief collapsed in 2012. Archaeologists, guided by years of research, began an excavation under a council car park in Leicester, a place where records suggested the friary once stood. Few thought they would find anything of value, let alone the remains of a king. Yet only days into the dig, they uncovered a skeleton in the area believed to have been the choir of the friary, the very spot where documents hinted Richard was buried. This skeleton, simply called Skeleton 1, held features that were impossible to ignore. It belonged to a man in his early 30s, the same age Richard was when he died. His bones bore clear signs of battle wounds, deep cuts to the skull, consistent with blows from swords and halberds, and damage suggesting his helmet had been torn away in his final moments. These matched the brutal accounts of Richard's death on the battlefield. Even more striking was the spine. The skeleton showed severe scoliosis, a curvature that would have caused one shoulder to sit higher than the other. Chroniclers from Richard's lifetime mentioned his uneven shoulders, while Shakespeare later exaggerated this into the image of a hunchback. The skeleton revealed the truth. Richard was not the twisted figure of legend, but he did live with a serious spinal condition that shaped how people saw him. Carbon dating confirmed the remains came from the late 15th century, the exact time of Richard's death. Chemical testing on the teeth revealed a diet rich in meat and fish, one fitting for a man of wealth and power. Combined with the burial location, the evidence strongly pointed to the remains being Richard III. But archaeology alone could not prove it beyond doubt. For history to accept the identification, scientists needed something irrefutable: DNA. Modern genetics offered a tool that previous generations of historians could never have imagined. By extracting DNA from the ancient bones and comparing it to that of living relatives, scientists could confirm the identity with remarkable accuracy. Richard III had no surviving children, so researchers had to look to his extended family tree. His mother, Cecily Neville, had many children, and through his sisters, living descendants could be traced. By following the female line, scientists identified people who carried the same mitochondrial DNA, which passes unchanged from mother to child across generations. They also studied the male line, examining the Y chromosome passed down from father to son. Richard himself had no surviving sons, but his broader Plantagenet family offered distant male line relatives, particularly those descended from Edward III. If the skeleton's Y chromosome matched theirs, it would confirm that the remains belonged to a true Plantagenet king. The discoveries that followed shocked the world. While the mitochondrial DNA confirmed the maternal link, the Y chromosome did not match the living male line relatives. This suggested something extraordinary, that somewhere in the centuries of royal lineage, an illegitimacy had occurred. A break in the bloodline meant that Richard's genetic signature did not perfectly align with those who claimed descent through the male line. The implications were staggering. Did this mean Richard's identity was in question? Or did it point to a hidden scandal that rewrote the legitimacy of dynasties across English history? The revelation left historians scrambling to reassess royal succession and the accuracy of centuries-old records. What had begun as a thrilling discovery beneath a car park had grown into one of the greatest historical controversies of our time, forcing us to question everything we thought we knew about Richard III, his bloodline, and the monarchy itself. The discovery of Richard III's skeleton in Leicester was already extraordinary, but DNA testing promised to turn the find into proof. The question was simple but enormous. Could fragile genetic material survive more than 500 years underground, and could it really confirm the identity of a medieval king? The task was daunting. Ancient DNA is extremely delicate, often broken into tiny fragments and at constant risk of contamination from soil, microbes, or even modern human contact. But if it could be recovered, it offered a kind of evidence stronger than any written record. The key was knowing which parts of DNA remain stable through generations. Most of our genetic material is reshuffled every time a child is conceived, half from the mother and half from the father. But two forms of DNA pass down the centuries with little change. Mitochondrial DNA, which travels from mother to child, and the Y chromosome, which goes from father to son. These became the central tools in the investigation. Mitochondrial DNA, or mtDNA, is inherited only from the mother. That means all children of the same mother share the same mtDNA, and it continues down the female line almost unchanged. Richard III had no surviving children, but his sister Anne of York did. By carefully tracing her female descendants through centuries of marriages and migrations, genealogists eventually found two living people whose maternal line led back to and Michael, a furniture maker in London, and Wendy, a genealogist in Canada. If the mtDNA in the skeleton matched theirs, it would be powerful evidence that the bones were indeed Richard III's. The paternal test was more complicated. The Y chromosome is passed from father to son, usually with little alteration. In theory, Richard III's Y chromosome should match that of other male line descendants from the broader Plantagenet family. The most reliable line traced back to Edward III, Richard's ancestor. From him came the Somerset family, descendants of Henry Somerset, the 5th Duke of Beaufort. If their Y chromosomes matched the skeletons, it would strongly confirm that the remains belonged to a true Plantagenet king. Extracting DNA from bones buried for centuries required extreme care. The remains were handled in laboratories designed to prevent contamination. Scientists wore full protective suits, masks, and gloves. They drilled into the teeth and dense bones, areas most likely to preserve DNA, then painstakingly extracted tiny fragments of genetic code. These fragments were fragile, often only a few dozen base pairs long, so machines had to copy them millions of times to create sequences long enough to analyze. The process was like reconstructing a shredded document, piece by piece, while making sure that no modern DNA had slipped into the mix. When the mitochondrial DNA results came back, the answer was stunning. The genetic code from the skeleton matched both Michael and Wendy. The odds of such a match happening by chance were astronomically small. For many, this seemed to confirm that the bones under the Leicester car park really did belong to Richard III. But then came the Y chromosome test, and with it a shock. The skeleton's Y chromosome did not match the Somerset family. Instead of confirmation, it revealed a break in the paternal line. Somewhere in the long chain of fathers and sons stretching from Edward III to the present, a recorded father was not the biological father. Scientists call this a false paternity event, a polite term for a moment when an illegitimate child was accepted as legitimate in the family tree. This discovery raised unsettling questions. Did the break occur in Richard III's own line, casting doubt on his place in the Plantagenet dynasty? Or did it happen in the Somerset line, meaning that some of today's descendants were not truly connected by blood to Edward III? With so many generations between the 14th century and today, the exact point of the break could not be identified without more genetic testing from other royal remains. What was certain was that the supposedly unbroken thread of royal blood was not so unbroken after all. The idea of sacred continuity in the Plantagenet line, once treated as the foundation of dynastic legitimacy, was revealed to be more fragile than many had ever imagined. For scientists, the match in mitochondrial DNA was enough to say with strong confidence that the skeleton was indeed Richard III. But for the public, the Y chromosome result became the most fascinating part of the story.